Hi, welcome to my presentation on kind of the steps that we follow when we are doing science and some considerations we have to keep in mind when we are trying to learn about things using science or use science to investigate the world. So first, I just want to go over the scientific method again. So of course, the scientific method is going to start with observations. So just, kind of something that you have noticed about the world around you that you are curious to know more about. So those observations that you're curious about lead you to have kind of a question about them. So the second step is to have a question that could be like, why are these things occurring? What makes them happen? Why is it that they happen the way they do? What effect do they have on something else? Anything like that could be a valid question. Once you have your question made that you're trying to figure out an answer to, first you want to start by making kind of a preliminary answer almost, which is your hypothesis. So the hypothesis is a statement that is basically explaining an answer to your question. It's based on your pre-existing knowledge about the world. Also, you would probably do some research into scientific literature and what type of experiments have already been done, what information is already known that is relevant to your question that would help you form a hypothesis. Then your hypothesis also, you just have to make sure that it is like testable and falsifiable, which means that it's possible to design an experiment to test the hypothesis, and it's possible to prove the hypothesis wrong. So once you have your hypothesis, the next step is to design that experiment that you would use to test it. So you're going to do an experiment, and of course every experiment needs to have control groups so you can make sure that it is working correctly, and they will also help you interpret your results from the experiment. So you would do the experiment, then you get your results, your data. So those are also more observations. So you observe what happens in that experiment, and that's your data. Then you're going to interpret that data and use it to either accept or reject your hypothesis. So if the data are kind of in line with your hypothesis, you could accept the hypothesis. If the data don't make sense with the hypothesis, then you need to reject the hypothesis. So if you had rejected that hypothesis because the data didn't support your hypothesis, you could just kind of like leave it there and start over from scratch with a new hypothesis, or you could take the hypothesis that you had to reject and just modify it to be a bit different and then do the experiment again. Now you're testing the modified hypothesis though. So you might do the exact same experiment, or maybe you need to do like a little bit different experiment to test the new hypothesis that you have. And you would just repeat the cycle, get your observations from that experiment, and interpret the data, see if they support or don't support the hypothesis. You could then accept or reject it again. On the other hand, if you did have data that supported your hypothesis, then you could accept the hypothesis, which means realistically at that point you probably want to write it up and publish it. But, as far as the overall project of science goes, what happens next is that you or maybe a different scientist is going to test that hypothesis again. So you're going to do the experiment again, maybe the exact same one to make sure it comes out the same way, or maybe you're going to figure out a different experiment that you could use to test the same hypothesis. So then you would have multiple lines of evidence that are all supporting that hypothesis, assuming that the new experiment you do, comes out with data that supported the hypothesis. Then, of course, once the hypothesis is very well supported by lots of different experiments, then it could be accepted as a theory or a law. So first, well, I guess second, we'll go over kind of the differences between a hypothesis and a theory in science. So a hypothesis, as I mentioned, is a testable and falsifiable statement that is explaining an observation or answering the question that you had about that observation. So the fact that it's testable, that means that it's possible for you to make an experiment that will, test if this hypothesis is true or not true. And falsifiable means that it is possible to prove that the hypothesis is wrong. So if it's not possible to demonstrate that your hypothesis is wrong, then that means it is not a valid hypothesis, and you would need to make a different one. So for example, you could have a hypothesis that says the Earth is flat. That is testable and falsifiable. So you could test that by building a rocket ship and going to the moon and looking back at the Earth and seeing that, seeing what shape it is, and you can notice that it's wrong. It's not flat. It's round. And at that point, you would notice that your hypothesis is wrong, right? So it's possible to falsify that hypothesis. Another example of a potential hypothesis might be magic mushrooms have an intangible, unobservable spirit living within them. So that actually is not a valid hypothesis because it is not falsifiable. Since the spirit that you propose lives in the magic mushrooms is not observable, that means it's not possible for you to demonstrate that it is not there, which means that that is not a falsifiable hypothesis, therefore it is not valid. So you would need to come up with something different that could be falsified. So that's a hypothesis. Then you have theories also, which kind of in day-to-day use, a theory means just kind of something that you think might be going on. It's like kind of speculative. It's a speculation that you're making or a proposal that you're making, but you're not sure if it's correct. But in science, that's not what we mean by theory. So a theory in science is an explanation that is supported by lots of different experiments and lots of different evidence. So it's basically proven to be correct. It's basically like a hypothesis that has been proven to be correct in experiments or has been supported by experiments over and over and over and over again, lots of different experiments. And so at some point we say that, this is probably just right. It's probably right. We don't say that it's actually literally proven. It's mostly proven, or it's kind of like almost proven. But we'll never say that anything is actually 100% proven in science because we always want to leave the door open for us to be wrong about something. It's always possible that we don't have all of the relevant information. It's always possible that in the future new technology could be developed that gives us the ability to do new experiments, and those experiments could turn up like new information that we weren't aware of before. That might contradict with our existing theories and things that we consider to be basically correct. So we're always acknowledging it's possible that we're not like right about things, and we never say that we have the final right answer in science. So we'll never say that something is proven. But a theory in science is like as close to proven as something can get without you saying that it's literally proven. Usually a theory is also going to be kind of more broad, and a hypothesis is going to be really narrow. Because a hypothesis is something really specific that you're trying to test, and a theory might be kind of, just not quite that narrow, not quite that specific. So as you're testing a hypothesis multiple times, you might kind of end up broadening it into a theory if it's always turning up correct. So just like we had just talked about how a hypothesis has to be falsifiable in order to be valid, there's actually only certain types of questions that are kind of valid to try to answer by using science and the scientific method. Questions that are valid to answer using the scientific method are any question that you could answer by observing something or by measuring something. So it has to be something that is kind of observable or measurable in the world around you. So that kind of relates to how a hypothesis has to be falsifiable. That means that it has to be something that, could be answered by, could be like, I guess, supported or rejected based on observations. Similarly, a question has to be something that can be answered using observations or measurements about the world. So there's some questions that you can't really answer using the scientific method. For instance, if you're asking, what is the meaning of life? That's not something that you can answer just by observing and measuring things. It's more of kind of an abstract question that doesn't have like just a simple right or wrong answer, or a simple observable answer that is kind of verifiably correct. So that means that we can't answer that question using science. Stephen Gould is an evolutionary biologist who has tried to kind of reconcile, I guess, science with religion. And so his idea for how they can be reconciled is non-overlapping magisteria, which means that basically science has its place and religion has its place, and they don't meet. So they don't overlap with each other. Each one is a magisterium, which I guess kind of just means like a realm of thought or realm of inquiry, and they don't overlap with each other. So basically they don't have that much to do with each other. So scientific questions would be things that you can observe or answer by observing and measuring in the world. So those should be answered using science and not religion. Questions about the meaning of life could be answered using religion if you're religious, but they wouldn't be able to be answered using science, and you shouldn't try. So I'm not going to say that that is like correct or not correct. That's just Stephen Gould's idea. But I guess there are a lot of people who kind of accept that idea, including a lot of scientists. So there's a lot of religious scientists out there actually, and they just kind of keep those two areas of their life a little bit separate from each other. So once you have your hypothesis, you would do an experiment to kind of investigate whether that hypothesis is correct or not. An experiment is something that you're doing in controlled conditions to test that hypothesis. You're going to be choosing one variable to manipulate in the experiment, and then everything else, every other variable, you're going to be controlling it so that it remains the same. And then you're going to be looking for the outcome and measuring that outcome. Then you're going to see if that measured outcome kind of matches with what you expected to measure based on your hypothesis. So you're going to see if it matches with predictions that you made in your hypothesis. Usually we do experiments in science, but sometimes there's something where we can't really actually control all of the conditions. In that case, we're not going to do like a proper experiment, but we still want to investigate maybe our hypothesis. So we're going to use an observational study. In an observational study, you're not actually using controlled conditions and you're not changing anything. There's not any variable that you're manipulating. You're just very, very closely watching everything and measuring and recording everything that you see. So an example of that would be like if you're trying to find out something about how a particular species of animal mates in their natural environment. If you're going to actually investigate that, you can't do it in an experiment because an experiment does not happen in a natural environment. It happens under controlled conditions. So you're not going to be able to perfectly mimic a natural environment during an experiment. So to figure out, the mating habits of the species that you're interested in, what you need to do is go into the wild and find, individuals of that species or a population of that species and just watch them kind of quietly, try not to impact them in any way. So ideally, they should not know that you're even there. So you're trying not to impact anything in this observational study. You're trying to just record without manipulating anything. So you can see what happens under natural conditions. So that's very different from an experiment where you are specifically using control conditions and you're trying to control everything in the environment. So for instance, if you're doing experiments that involve animals, that would mean that you need to be doing that experiment always at the same time of day, at the same temperature, the same humidity, the same level of light. Those animals need to be fed at the same time of day. They need to have the same lights on, lights off time. Everything related to those animals must be controlled and kept absolutely constant during the experiment so that those other variables can't impact the results of the experiment. Also, experiments need controls. A control is different from controlled conditions. Controlled conditions means that everything that you can think of, you're trying to hold constant, except that one variable that you are manipulating in the experiment. A control, though, is another basically group of individuals that you're testing in the experiment, but they're not going to be tested in the same way as the others. So you're going to be using this control to just make sure that your experiment is working correctly, to make sure, like, if there's any variables that you weren't able to hold constant, that they're not impacting your results, and they also help you interpret your results. So an example would be like if you're doing an animal study where you want to see if a particular drug impacts the way that animals eat their food, you would need a control where you have a group of animals that don't get the drug at all. So you're going to have one group of animals where they get the drug. Maybe you might have multiple groups and they get like different concentrations of the drug or different doses of the drug, and then you're measuring how much they eat or what different things they eat. And you also need to have your control condition, which is the group of animals that didn't get any drug, and you're also measuring how much they eat, just to make sure everything is kind of going normally. And so that you would be able to say, like, when the animals got the drug, they ate more or they ate less. You're comparing to the control condition where the animals ate at a normal level because they didn't get a drug. And then kind of a final point to keep in mind when designing an experiment is that usually you want to have kind of as many individuals involved in the experiment as you can, just to make sure that weird things aren't going on, or that if weird things are going on, they're not impacting the results in a major way. So of course everybody's different. That applies to animals as well. That even applies to cells. They're all a little bit different from each other. So if you're testing something and you're using, only one animal, like let's say, you're testing how a drug affects how animals eat. If you have just one animal who got the drug and you're measuring how that guy eats, then you're not measuring how like animals in general eat or animals of that species eat. You're just measuring how that individual animal is affected by the drug. Different individuals could be affected in different ways. So you want to have multiple that you're testing. In general, just the more, the better. Yeah, you kind of end up limiting it because like the more animals you have or the more individuals you have, even if it's just cells, the more you have, the more expensive it is to do the experiment, and it kind of just turns into a bigger setup that you have to have. But at the same time, you want to have as many kind of as possible because that gives you just better results. It lets you know that you can kind of rely on your results to be accurate, to a greater extent. And then at the end, when you're trying to do statistical analysis of your results, it's easier to do the statistics, and the statistics are more reliable if you have more individuals in your experiment. So there's different types of variables and controls that we use in experiments. And there's two basic types of variables and then two basic types of controls that every experiment should have, or that almost every experiment should have. The variables are the independent variable and the dependent variable. The independent variable is the variable that you are changing in the experiment, so it's the thing that you're manipulating. The dependent variable is the variable that you're measuring, so that's like the outcome that you're measuring in the experiment. Any other variable that's present, you should be holding it constant, and it should be a controlled condition. Then for controls, the two most basic controls that you would have in almost any experiment are a negative control and a positive control. The negative control does not show the outcome that you're looking for in the experiment, so it does not show the thing that you're expecting. The positive control does show the outcome you're looking for, or the thing that you're expecting to see in the experiment based on your hypothesis. So for example, maybe you're doing an experiment where you have cancer cells and you're exposing them to a particular chemical, let's say the chemical mangosteen, and you want to see if this chemical is toxic to the cancer cells because you think that maybe this chemical could be used as like a treatment for cancer. So you want to see if we put the chemical mangosteen with the cancer cells, like, do they die or do they live? If they die, maybe it's a potential cancer treatment. So you've got your two variables here. The first variable, the independent variable, is the mangosteen itself. How much mangosteen are these cancer cells being exposed to? That is the independent variable, or the thing that you are manipulating in the experiment. The dependent variable is the thing that you're measuring, which in this case is cell death. So you're thinking that this chemical mangosteen may kill cells, so you apply it to the cells and you want to see, do they die or do they live? So the thing you're measuring as your dependent variable is the cell death. Then you're going to need to have two controls, your negative control and your positive control. For this experiment, your negative control would be cancer cells that do not receive any treatment. So you don't put mangosteen with them, you don't put anything with them. That's to see how these cells are going to live under normal conditions. So you want to know kind of, how many cells would die naturally, just, because cells die eventually. So, anytime you have a dish of cells, like some of those cells are going to be dying just because basically of old age. So when you treat your cells with mangosteen and you see that some of them die, you want to make sure you know that like they died because of mangosteen, they didn't die just because of old age. So that's why you have to have your negative control. It helps you interpret your results. You need to be able to look at your mangosteen treated cells and see that they died at a higher rate than the cells that got nothing at all in the negative control. The other purpose of the negative control is just to make sure that your experiment is done correctly. You have conditions under which cells can, live. So if the cells all die in the negative control and you didn't treat them with anything because it's a negative control, that lets you know that like you are not caring for those cells correctly. So then when you see the mangosteen treated cells die, like that's not because of the mangosteen, that's just because I don't take care of cells good. All my cells were going to die anyways. So you would need to kind of redo that experiment and figure out what it was that was killing your cells. Something about the way that you were maintaining them wasn't right. Maybe they didn't have the right nutrition or something like that, and you need to fix that. And then you need to see that your negative control is coming out with cells that are alive. Basically alive, with a few that are dying of natural causes. So if your control doesn't look the way you expect it to look, that means that something went wrong in your experiment. It's not designed correctly, or maybe it is designed correctly, but you didn't execute it correctly. Something's going wrong. So you have to fix it and redo it. If your control is going the way you expected, then you get to use that control as a basis for comparison to help you interpret your results. So you get to say 5% of cells die in the negative control of natural causes. In the mangosteen treated cells, 15% of them died, so that's more than we would expect to die of natural causes. Therefore, mangosteen does actually have some toxicity against cancer cells. So that's the negative control. Then you would also have the positive control. So in this case, your hypothesis is going to be that mangosteen does kill the cancer cells. So the positive control is the outcome that you're looking for. It should show the outcome that you're looking for. So what you want to see there is cell death. You're looking to see the mangosteen kill the cancer cells. So in your positive control, you need to find some way of killing the cells so you can show cell death in that control. So what you would do is find some substance that you already know for a fact is toxic to cancer cells and apply it to those positive control cells. Actually, we might use a drug that is already used as a cancer treatment because we know it's going to be toxic to cancer cells for sure. So you treat the cells in the positive control with that known poison, and then you should see that they die. If you're not seeing them die, well, things aren't going right then. You need to figure out what's wrong and fix it. So, if the positive control doesn't turn out the way you expected, that lets you know that your experiment is not going right. Something is wrong with it. For instance, in this case, it might be that you're just not able to tell the difference between a living cell and a dead cell, right? So, I mean, dead cells actually don't look that different from living cells. They look like little see-through bags. So you need to have some kind of way to make sure you know that they're dead. So if you apply a cancer drug to some cells and you don't see them die, probably they did die. Probably you just don't know how to identify dead cells. So, by using the positive control, you can make sure that you have some way of knowing for sure that the cells do die, when they die. For instance, you would probably use some type of stain that only stains dead cells, and then you should see all the positive control cells kind of lighting up with that stain, and you're like, oh, those are dead, and I can tell what dead cells look like now. So now, when I go to look at my mangosteen-treated cells, I can see them die. So, let's say that you did your experiment, maybe on those cancer cells, giving them the mangosteen, and they didn't die. And you looked at your positive control and you saw a bunch of dead cells, so that's good. You looked at your negative control, you saw a bunch of living cells, so that's good. Since your controls turned out the way you expected, that lets you know that your experiment was designed correctly and you also did it correctly. So your results should be valid. and your results were that the mangosteen did not kill the cancer cells. So now you have to accept that, your hypothesis was wrong. You have to reject it, and you have to kind of like accept that rejection. So sometimes scientists will get kind of, attached to a hypothesis, and they won't really want to reject it when their experiment shows that it's wrong. And of course, that's just a feature of people in general, right? We don't like to be wrong about things, and we don't like to admit that we're wrong. We would prefer to find kind of a way that we could be right, actually. But if in science, if you're going to be, a good scientist, you have to be able to admit it when you're wrong. You have to kind of let go of that attachment you have to that idea that you really liked, and just admit that, that idea was wrong, if it comes out as not being supported by your experiment. So for an example of this, on Netflix they have a documentary called Behind the Curve, which is about flat earthers, which is people who believe that the earth is flat and that there's kind of a giant conspiracy to make everybody believe that it is round. So some of these flat earther people will do actual experiments to try and prove that the earth is flat. And of course, when they get the results do not support their hypothesis that the earth is flat, but in general, they do not, they don't reject their hypothesis. They don't accept their results. Instead, they say that, something must have been wrong with the experiment, just something went wrong somewhere, the earth is flat, and we know that, so it doesn't matter what these results say. So a quote from that documentary I have a quote in here from this documentary, from a guy called Bob Knodel. So he and a group of flat earthers had actually pooled their money and bought a really expensive gyroscope, a multi-$1,000 gyroscope, and it's really, really sensitive. So over here you can see a basic gyroscope. Gyroscopes are designed to measure acceleration. So actually inside of your phones, a lot of you will probably have a little accelerometer, which is a little gyroscope that helps the phone kind of just know how you're moving it and how it's positioned. This is actually an example of the gyroscope that they bought. So, it's a very, very sophisticated, extremely sensitive gyroscope. It's actually so sensitive that it can sense the rotation of the Earth around its axis. So as the Earth is rotating, that's going to be picked up by the gyroscope as drift. And so they were buying this gyroscope and using it to measure the drift of the Earth, and they were expecting to not find any drift, because if the Earth is flat, it doesn't rotate, and you're not going to find, you're not going to find that drift. You would only pick that up if you have a round rotating Earth. So Bob Knodel had this to say about the outcome of that experiment. He said, quote, "What we found is when we turned on that gyroscope, we found that we were picking up a drift, a 15 degree per hour drift. Now, obviously we were taken aback by that. Wow, that's kind of a problem. We obviously were not willing to accept that, and so we started looking for ways to disprove it was actually registering the motion of the Earth. We don't want to blow this, when you've got $20,000 in this freaking gyro. If we dumped what we found right now, it would be bad. It would be bad. What I just told you was confidential." So this guy, he did his experiment to show that the Earth is flat. They obviously spent a lot of money on this gyro. They had an investment, kind of a psychological investment, and proving that the Earth is flat. And at that point, they also had a financial investment in it as well. But that's not what the results of their experiment showed. And at that point, what they should have done as scientists is accepted the results of the experiment and, rejected their hypothesis that the Earth is flat. But that is not what they did. Instead, they decided to try to conceal these results. I'm not sure why he decided to talk about it on a documentary when he was being filmed. But they decided to try to conceal the results until they were able to get more results that would disprove that the gyro had actually picked up the motion of the Earth, which of course they're not going to be able to do because, the Earth is round and it rotates. So at that point, that's when they stopped doing science. This started as an experiment, but when they failed to accept the results of their experiment, that's when they stopped doing science and started doing a conspiracy theory instead. So just, we all hate to be wrong as people, but in science we have to kind of get over that and look past it and be able to accept the results of our experiments even when we don't like them. So now we'll just go over a couple of examples of kind of science in action and how the scientific method is actually used to investigate questions. So first, an example involving giraffes. So here the observation is that giraffes have really long necks, and the question is, why is that the case? So I guess I actually kind of missed putting observation on this slide as the first thing, but the first thing is you notice giraffes got a real long neck. That's funny. And then the second thing is you say to yourself, why do giraffes got such a long neck? And that's your question. So that is a question that you can investigate using science. So your next step is to make a hypothesis about that. So of course, the kind of most obvious explanation that springs to mind is that giraffes have a long neck because that lets them get food that's really high up in the branches of a tree, so that while other animals are stuck kind of grazing lower in the branches, giraffes can get the stuff at the tippy top of the tree, and that helps them compete for food. In that case, you would start with kind of giraffes that have more of a normal sized neck, and you would have a few that just had by chance a longer neck, which helps them get higher leaves, so they are able to get more food, and that gives them a reproductive advantage over other giraffes. It increases their fitness. Since they can get more food, they can have probably more children, and over time, they pass that longer neck onto their children. Over time, you get giraffes with longer and longer necks, since it's always an advantage to have a longer neck because it lets you reach more food. So that's the hypothesis here. The second, or I guess I should say the next step, is to just check your hypothesis, make sure that you stated it really clearly. There's not any confusion in the language that you used. Make sure, of course, that it is testable and falsifiable. So you have to find a way to test that hypothesis, and it has to be possible to demonstrate that your hypothesis is wrong. After you have kind of checked the validity of your hypothesis, next you're going to list the predictions that come from your hypothesis. So if your hypothesis is correct, if you assume that it's correct, what would you predict to be the case? So for instance, you might predict that different giraffes will have different neck lengths. So neck length is variable. You might predict that giraffe neck length is heritable, so when a long-necked giraffe has children, they pass on that long neck to their children. And you might predict that giraffes that have the longer neck will eat more food than giraffes that have a shorter neck. And you might predict that giraffes will prefer to eat food that's higher up in the trees rather than lower in the trees. So with all these predictions, we could say that, if giraffes have necks of different lengths, and if neck length is heritable, and if longer necks lets you eat more food, so that's an advantage, then you're going to have evolution occurring by natural selection over time to drive a longer and longer neck length. And you should also be able to observe, the giraffes that have the longest necks eating the most food, and, giraffes in general liking to eat food that's higher up in the tree. So once you've got your predictions down, then you can make what's called a null hypothesis. The null hypothesis is kind of predicting what would be the case, or what would happen if your regular hypothesis was actually wrong. So if your regular hypothesis is predicting that giraffes that have longer necks eat more food than giraffes with shorter necks, your null hypothesis would say that giraffes that have longer necks will not eat more food than giraffes that have shorter necks, so they'll eat the same amount of food. If your hypothesis predicts that giraffes prefer to consume food higher up in the trees, then your null hypothesis would state that giraffes do not prefer to consume food that's higher up in the trees. So now you've got all your stuff with your hypothesis worked out. You have your predictions that you can test to see, and if they come true in an experiment. You have your null hypothesis, which is just basically kind of the opposite of your hypothesis. Then you're going to finally design an experiment to test this hypothesis. Now in this case, since we're looking at giraffe neck length, and we're trying to see how it affects their ability to consume food, because we're trying to figure out if it helps them consume more food or higher up food if they have a longer neck, that's probably going to be something that we answer using an observational study. It's going to be just kind of hard to do an experiment on that. There's a lot of variables to control, and also you kind of want to know like what these giraffes are doing naturally under the normal conditions that they live in. So if you put them in a lab, first you're going to have a really big lab to house a giraffe and some trees, but also, that's not a normal environment for the giraffes. Maybe they're not going to behave the way that they normally would, and you want to see them behaving normally. So in this case, we're actually not going to make a proper experiment, we're going to make an observational study. So for instance, maybe you could go out into the wild and find some giraffes and just measure their neck lengths and then measure how much food they eat, and try to see if there's a relationship there. Or maybe you could go out into the wild and look at giraffes when they're eating and kind of measure like what height they were eating at. So that's an observational study that was actually done by a couple of scientists called Young and Isbell, and they published it in 1991. So we're going to be kind of talking about the observational study that they did, measuring the height that giraffes prefer to eat at in the wild. So once you have your experiment designed, you just want to make sure that it does have controls, if appropriate. Sometimes in an observational study, you might not really have like controls properly because you're not manipulating anything in an observational study. So it might not be appropriate to have controls in an observational study. But usually you would need them. In this case, for our observational study, you would just want to make sure that you're testing or you're observing different types of giraffe groups. Not just picking one group that you look at, but you're looking for different types of groups. So sometimes, giraffes will live in a group that's like all males, or you might have a group that's mostly females, but it has a couple of males. So they have different group compositions. You would want to make sure that you're looking at different group compositions so you can see kind of on average how they all eat, and you're not looking at, something that's just determined by the group composition. So then you would actually, go out and do that experiment or do that observational study, get your data from it, and then finally you need to interpret that data. And based on the data, either accept or reject your hypothesis. So these are a couple of graphs that Young and Isbell made and published in their 1991 study, and showing what height male giraffes like to eat at. So on the y-axis here, you have the proportion of bites they took at different heights. On the x-axis, you have different heights of trees. So this is measured in meters. So this is 1 meter, 2 meters, up to 7 meters, which would be pretty high. That's like, roughly 21 feet high, plus or minus a bit, plus a bit. And then here are the proportions. So what we're looking at here with this black dot at the 3, that means that basically 30% of all the bites that these males were recorded to take occurred at a height of 3 meters. So at the 6 here, you have this little dot, and that's roughly at the level of 0.1. So that means that about 10% of all the bites they observed being taken were taken at a height of 6 meters. So this is kind of like a histogram in a way. Then they also looked at feeding rate, so I guess just kind of how quickly those males are eating, and that's graphed using these open circles. So again, at the 3, you have this kind of open dot. That means they were eating at a rate of 0.45. I'm not sure what the units are there, to be honest, at a height of 3 meters. So this is just helping you compare like what rate giraffes were eating at and how many bites they took at different heights in the trees. And this is for male giraffes that are in groups that are just males, or male-dominated groups at least. So you can see that actually most of the bites that are taken are at a height of about 3 meters, and then it kind of falls off from there. So they like to eat at about 3 meters. Also, they like to eat, a little less at 2 meters and at 4 meters, but as you go further from 3 meters, they don't like to eat there as much. And the same thing is kind of shown with the feeding rate. They eat fastest at a height of 3 meters, and then it kind of drops off on either side from there. So their preferred eating height seems to be kind of, 3 meters high, which is, that's high, but it's not crazy high. For a giraffe, that's actually about shoulder height. For a male giraffe, that would be about shoulder height. Then they have this other graph down here, which is showing the pattern for male giraffes in a group of female giraffes. So these would be the dominant males in a group of mostly female giraffes. And there you can see some difference. So these giraffes like to eat at a height of 5 meters. About 40% of all the bites that were taken were at a height of about 5 meters. And then it gets kind of, it goes down from there at heights that are above that or below that. So if you're kind of looking at that data and relating it to the predictions made by the hypothesis had predicted that giraffes will prefer to eat food that's higher up in the trees. But actually, we're seeing that males and groups of all males, they prefer to eat food that is shoulder height for them. For males that are in female groups, they actually prefer to eat a bit higher, but the average male giraffe is actually 6 meters tall. About 3.5 meters at the shoulder, but 6 meters tall overall. So these males in the female-dominated groups are actually still preferring to eat leaves that are a bit shorter than they are. So 5-meter high leaves, not 6-meter high leaves. So that hypothesis is not being supported by these data. So based on kind of the overall impression that we're getting from the data, we're going to have to reject the hypothesis that giraffes have long necks because that helps them eat more food since they can eat higher in the trees. So we're going to instead have to accept the null hypothesis that giraffes do not prefer to eat food that's higher up in the trees. So since we just rejected our hypothesis, what we got to do next is like make a new hypothesis, and then we could test that. So that's what's going on here. We're going to make a new hypothesis for why giraffes have such a long neck. We're going to call this an alternate hypothesis because it's not the one that we kind of started out with. Then we're going to have to do a new experiment or maybe observational study to test the new hypothesis. So for instance, one potential alternate hypothesis you might make is that giraffes have a long neck because that helps them fight. It helps the males fight over females to get kind of mating rights. So then if you have a longer neck, you do better in the fight and you're more likely to find a mate, and then that gives you a reproductive advantage because you're going to have more children when you can find more mates. So then longer neck giraffes would, have more offspring, pass on their long necks, and overall, over time, giraffes would evolve to have longer and longer necks. So once we have that new hypothesis, that alternative hypothesis made, we would want to go through the same steps with it. First, of course, we're going to check it to make sure that it's falsifiable and testable and, generally a valid hypothesis and clearly stated. Then we're going to list some predictions from that hypothesis and figure out a null hypothesis, and then figure out a way to test it using an experiment or an observational study. So one prediction that hypothesis might result in would be that, you might want to say that longer neck giraffes will mate more times over the course of their life than shorter neck giraffes. Or you might say that longer neck giraffes will be observed to win fights more frequently than shorter neck giraffes. Those could be some predictions that come out of that hypothesis. Then your null hypothesis would just be the opposite of those. So for instance, you might say that longer neck giraffes do not win fights more frequently than shorter neck giraffes, or longer neck giraffes win fights at the same rate as shorter neck giraffes. Then you could do an experiment or an observational study. Again, probably it'll be an observational study again because we're looking at giraffes to test that. So maybe you would go out into the wild and observe groups of giraffes where males are interacting and just watch them fight and just record the neck length of giraffes that win the fight. Something like that. So this actually was done in another observational study, and it turns out that giraffes that have a longer neck do win fights more frequently. I guess when giraffes are fighting over a female, they will kind of swing their necks at each other, and they're kind of using their necks as a club. And just the longer the neck you have, kind of the more speed you can get, the more force you can get going behind your club, and so it's a more effective weapon, basically. So longer neck giraffes do, win fights more frequently than shorter neck giraffes, and that's probably, our at least best idea right now of why giraffes have ended up with such long necks. At the conclusion of your study, actually, whether it is, kind of the first study, Young and Isbell's study, or the new study that's looking at, you know, giraffes fighting over females, either way, once the study is over, you gotta publish that. So the last step is to communicate the results of your experiment or your observational study to the larger scientific community and let them know if your hypothesis was supported or invalidated. That way you are contributing to kind of the general progress of science, and other scientists can use your work to make their own hypotheses and refine their own work. So you could communicate those results by doing a presentation at a conference. So different scientific disciplines will have different conferences, throughout the year in different parts of the world. So you could travel to one of those conferences and present maybe a poster about your study or actually give a talk about it to other scientists. But kind of the most common way to communicate results of an experiment would be to publish a paper about it. So you would actually write up your experiment as a paper, with an introduction, methods, results, conclusion, and you would publish that in a scientific journal where it would be peer reviewed. So other scientists who work in a similar field would read over the paper and just make sure that your methods are valid, your experiment was done in a valid way, and that the results you're reporting support the conclusions that you have made in the paper. And so that's kind of the way it's supposed to go. You have to communicate results somehow, or else it doesn't really matter that you did the experiment. Only you will know, and it doesn't help science at large if you don't communicate those results. In reality, there's some issues with this. Because usually scientific journals like to publish experiments that where the hypothesis was supported. Where the hypothesis was kind of demonstrated to be accurate. A lot of journals don't like as much to publish experiments where the hypothesis was rejected. That's kind of seen as an interesting outcome. So that's not really a valid perception. That's just kind of our human bias influencing our science, and it is unfortunate, but that is kind of the way things are working right now, unfortunately. So we'll look at just one more example of kind of the scientific method in action in a study that involved ants this time, and is like an actual experiment, not an observational study. So this study is about Saharan desert ants, which are ants that live in the Saharan desert, and observations that people have made about these ants are that when they're looking for food, they just kind of wander around the desert at random until they find it. But when they find some food, then they kind of make a beeline straight back to the nest. And until they get like pretty close to the nest, and then they start making like a lot of U-turns until they actually find like the entrance to the nest. So that's kind of strange. They're just kind of wandering aimlessly around the desert, but then they know exactly like how to get back to the nest using a straight line. So the question in this case would be like, how do they do that? Like how are they navigating back to their burrows? And so from previous research on these ants, we know that they are using the sun as a compass. So that's how they know like what direction to go back to their nest, but they don't use landmarks. So they're not keeping track of like big rocks that they passed or like a particular shape of a dune or anything like that. So if they're using the sun as a compass, that lets you know like how they know like what direction they should go in, but how do they know like when to stop walking in a straight line? How do they know when to start doing the U-turn so they can make sure they find the nest hole? So the question for this study is just that, like how do the Saharan desert ants know when they get close enough to their burrow to start doing the U-turns? So then after you have the question, the next step would be, of course, make a hypothesis. So these scientists said that they know when they're close to their burrow because they can remember like what angles they walked at as they were walking, like as they were wandering around. They remember what angles they were walking at compared to the sun. and they remember how many steps they took, and they know how long their strides are. So they can kind of synthesize all of that information to figure out how far from the nest they had gone, and therefore how far they need to walk in a straight line back to the nest until they're pretty close to it. So after you've made the hypothesis, the next step, of course, is to kind of double check your hypothesis, make sure it is stated clearly, it is testable, it is falsifiable, so that it's a valid hypothesis. Then once you've double checked your hypothesis, you can list some predictions that your hypothesis makes, or that you can make based on your hypothesis. So one prediction would be maybe that the length of the ant's stride is affecting their ability to navigate, and another prediction might be that how many steps they have taken affects their ability to navigate. Then you could make a null hypothesis, which again is just the opposite of your hypothesis basically. It's the predictions that should occur if your hypothesis was not correct. So in this case, it would be either that stride length does not affect the ant's ability to navigate, or that step number does not affect the ability to navigate for these ants. And then this image here is just kind of showing the path that these ants might take as they're foraging for food. So here's their nest, and they start kind of wandering at random away from the nest, just walking in random patterns until they encounter some food. And when they get the food, then they go straight back to the nest in a straight line. And when they get close to the nest, they start tracking back and forth like this, making these U-turns back and forth until they actually encounter the hole that's the entrance to the nest. So now that you have your predictions from your hypothesis and everything like that, you would be ready to design an experiment to test that hypothesis and see if those predictions come true. So when we looked at the previous example with the giraffes, we didn't actually do like an experiment. It was an observational study, or rather, those scientists did an observational study, Young and Isbell. But in this case with the ants, we get to do an actual experiment where we manipulate a variable and then measure the results in controlled conditions. So in this experiment, which was actually performed by, in real life by the scientists Whitlinger, Weiner, and Wolf in 2006, or they published in 2006, they caught ants and kind of raised them in the lab, or kept them in the lab, and they had an opportunity to kind of be introduced to their new lab environment. So they had a nest in the lab, and then there was like a trough that they could walk through to get to a feeder where there was food. And so they spent some time training on just getting food from that trough and bringing it back to the nest, and then they did the experiment. So in the experiment, they were actually changing the stride length for the ants, and they divided the ants into 3 different groups of 25 ants each. In group 1, they actually cut the legs of the ants so that they would be shorter. In group 2, they did nothing to the ants, and in group 3, they glued bristles to the legs so that the legs would be longer. So here you have images of those 3 groups. Here's group 1 with the short cut legs. And here's group 3 ant with these red bristles glued onto its legs so they're longer. And then in group 2, you have regular ants that were not messed with in any way. So of course, one of those groups is the control group, and it is group 2, the group that had nothing done to those ants. So in this case, that would be like a negative control because you're not doing anything to the ants. They're just kind of representing the natural state of those ants to give you like a basis for comparison. So after you've designed the experiment, your next step would be just to double check that you are using appropriate controls for the experiment. In this case, we do need a control. We need the group 2 control, ants that have a normal leg that is not manipulated, because we need to know how they're able to navigate back to their nest without anything being done to them to affect that navigation. So we want to see just standard navigation, and then that gives us something to compare the navigation for the ants that have weird leg lengths to. And in this case, we actually don't need another control for this experiment. Just the one is enough. So here is kind of an overview of their experimental setup. So you have this trough the ants had like a nest in the lab, and they had a trough to walk through to get to a feeder. And basically they trained them on this setup for a while, and then once they got used to it, then they did the experiment. So they would let the ants move from the nest and walk to the feeder. Then once they got to the feeder, that's when they would grab the ants and do something to their legs, or else not do anything to their legs. So for the group 1 ants, they grabbed them when they got to the feeder and cut their legs. For the group 2 ants, they grabbed them at that point and glued bristles to their legs. For the group 2 ants, they grab them and then just let them go. You still have to grab them in case grabbing them, affects the way that they're able to navigate. That way you're controlling for that variable, the potential impact of that variable. Then you let the ants go and see like how they get back home to the nest. So you know they're going to walk in the right direction, but when do they start making those little zigzaggy U-turns to actually find the nest? And you're able to actually measure exactly the point that that occurs at. And so we found that, here you have a graph of the results. Here's the group 2 ants, the normal ants, and you find that they start zigzagging to get to the nest at 10 meters out from the nest. So right here compared to the nest over here at the 20-meter mark. Then for the ants that had, the group 3 ants that had stilts or the bristles glued to their legs, they actually kind of overshot, and it was only 15 meters, or at about 15 meters is when they started doing their zigzags. For the ants that had their legs cut in group 1, they started doing the zigzags at only 5 meters. So here it looks like you are seeing an effect of the stride length on the ants' navigation. They went out to the feeder with the regular stride length, and that's kind of the calculation that they were relying on to get back. So then when they got back, when they were going back and they had longer legs, that gave them a longer stride, but they didn't account for that. So they went too far. When they were going back and they had shorter legs, their strides were shorter and they weren't able to account for that either, so they ended up not going far enough compared to the normal non-manipulated ants. So based on those results, they would accept their hypothesis that the stride length is affecting how the ants navigate back to their nest. And then finally, I guess I'll just mention the type of graph that this is. This is a box and whisker plot. So you're kind of showing the spread of the ants here. So let's actually I'll just talk about the normal the normal ants here. So they have a box. The line in the box is the median line. So that's like half the ants walked further than this line, half the ants walked less far than this line, and then you have quartiles. So at this line here, the edge of the box, you would know that 25% of ants stopped after that line and 75% stopped before it. At the other edge of the box, you would know that 25% of ants stopped before this line and 75% stopped after it. And then these whiskers are showing kind of the outliers. So, the ant that went furthest went this far, and the ant that went least far went this far. So the first part of the hypothesis, or I should say the first prediction, that the stride length is affecting the ant's ability to navigate, that's what we tested, or that's what the scientists had tested in that first experiment that they did. But they actually did another experiment to test the second prediction from their hypothesis, which was that the number of strides is also affecting the ant's ability to navigate. So that's going to be a similar experiment, but just a little bit different to test the different prediction. So in this experiment, they did it after the first one. So they've already got ants that have legs that are manipulated. So they're starting out with these 3 groups of ants with different sized legs. These ants are all going to start out at the nest, and they let them walk down to the feeder, and they basically then just let them walk back. So they let them walk back, and they see when do they start zigzagging to look for the nest. So the normal ants in the previous experiment had started zigzagging at about 10 meters. So they were doing kind of a normal calculation. They knew their stride length, they knew presumably the number of strides they had taken, and they were able to use that to calculate, when to or how to start zigzagging when they were 10 meters out from the nest. in the previous experiment, the other ants had their stride length manipulated, but they didn't really realize that, so they went either too far or not far enough. But in this experiment, we're curious to see what's going to happen, because they already walked out with their legs being, at that manipulated length, so they would be able to account for their stride length in their calculations. So maybe they're going to get back to this target of 10 meters out, starting to zigzag. And when we see the results graphed in a similar way, we see that it is the case. They actually all corrected the errors that they were making. So the normal ants started zigzagging at 10 meters, and the long leg ants started zigzagging at about 10 meters, and the short-legged ants started zigzagging at about 10 meters as well. So they're all basically starting to zigzag at the same point. So that means that when they are able to kind of know what their stride length is, then they're able to kind of get back to that original calculation of when they should start zigzagging, which would be based on their stride length and on their number of strides. So we kind of already went through this a bit, but of course after doing your experiment and getting your data, your next step would be to analyze that data and interpret it and see whether or not it supports or does not support your hypothesis. You're trying to see if the predictions that you made from the hypothesis kind of came true or not in that experiment. In this case, our data are supporting the hypothesis. It's showing that when we changed the stride length for the ants, they had a hard time navigating home the same way that non-manipulated ants would navigate home. So the ones that had their legs lengthened, they went too far. The ones that had their legs shortened, they went not far enough before they started doing the zigzags. So of course that's showing the effect of stride length on their ability to get home, but it's also actually showing that the stride number is having a role to play there as well, because the ones that had longer legs went too far and the ones that had shorter legs went not far enough. So it's kind of like they're assuming that their stride length is the same as when they set out from their from their home, and they just are remembering how many steps they took, roughly. So they're just trying to go back and take the same number of steps as when they went out, but now their steps are longer for the the ants that had bristles attached to their legs, so they go a further distance when they take the same number of steps. For the ants that had their legs cut, now their steps are shorter, so when they take the same number of steps, they're going to go a shorter distance. And then, of course, when you let them kind of just start from home with their legs being a weird length and then go to the food and come back, then they don't have a problem getting back home as normal. So they're able to kind of do that calculation, take into account their new stride length and the number of steps to start zigzagging at 10 meters just like a normal ant would do. So in this case, we get to accept our hypothesis and publish a paper. On the other hand, if the graphs had not supported the hypothesis, then of course you would need to reject it and make a new hypothesis, do a new experiment. So of course the really important part is that last part where you publish it. Well, I guess I should say all the parts are important. You can't really skip any. Except maybe the null hypothesis, to be honest, but apart from that, they're all important steps. But the part that is really kind of your bread and butter is the part where you communicate the results at the end by publishing a paper or at least presenting the data at a conference. Otherwise, no one's going to know that you did this and no one's going to care. So that's kind of a basic overview with a couple of examples of how science works in practice, especially in biology.